Hello, welcome to lesson 7.5. This lesson is about mycophagous nematodes, which we also call fungivorous nematodes or fungal-feeding nematodes. And this lesson is particularly about how these fungal-feeding nematodes interact with their fungal food source. We will also look at some studies proposing that fungal-feeding nematodes can be used against what we call plant disease, how the various fungi associated with plant disease can be controlled by the introduction of fungal feeding nematodes. Okay, let's begin lesson 7.5. Fungal feeding nematodes prefer the presence of fresh organic matter in the soil, which supports fungal diversity and therefore active biomass of fungi, which the nematodes can feed on. They influence fungal diversity, abundance, and community structure, and have specific grazing preferences that depend on the fungi present in the soil, as well as the grazer biodiversity, so the diversity of the fungal feeding nematodes. As they feed on fungi, they excrete excess nitrogen into the soil and therefore automatically increase microbial activity. As that excess nitrogen in the form of ammonium is excreted by fungal feeding nematodes, immediately that becomes available to fungi and bacteria that grow in the soil, increasing microbial activity. And this, of course, increases crop growth as well, as that ammonium becomes available to the plants. So in effect, fungal feeding nematodes increase bacterial foods and therefore bacterial populations. And the movement of nematodes in the soil increases microbial dispersal as some of the microbes move with the nematodes. It also increases oxygen supply to the soil and nutrients as these nematodes release those excess nutrients as they feed on fungi. So by default, all of this means that fungal feeding nematodes increase decomposition of organic matter, and that means that they affect carbon cycling, respiration of soil due to the increased microbial activity, and nitrogen mineralization. So they actually have a large effect on the soil ecosystem through their grazing behavior and interaction with the different components, both microbial as well as physical components of that ecosystem within which they reside. This is an image of a very common fungal feeding nematode called Aphelenchus aveni, and this character will feature in a lot of the future slides in this lesson. Some of the most common genera of fungal feeding nematodes include Aphelenchus, which we've seen in the last slide, Aphelenchoides, Ditilencus, and Tilencus. They pierce hyphae because they possess a stomatostyle or odontostyle, which is a stylet or a spear as part of their mouth parts. So here is one of these devices that they use in order to pierce fungal hyphae. The nematodes are usually polyphagous, so that means that they will feed on various types of fungi, including saprophytic or saprotrophic fungi, those that decompose organic matter, plant pathogenic fungi, so pathogens of plants, and of course mycorrhizal fungi as well. Some of them are quite specific in terms of their food preferences, and this depends on fungal susceptibility versus the resistance that some fungi possess in order to prevent being predated by fungal feeding nematodes. In the image on the right, we have an example of Ditilencus species showing its fungal feeding mouthpart. This video was generously provided by Dr. Harish Bajaj, an ex-professor and the head of the Department of Nematology at Ahasa University in India. And as you can see here, the nematode is piercing the fungal hypha with its stylet, And you can also see how the contents of the fungal hypha is being drawn through that straw effect. And you can see the pumping mechanism that the nematode has as well within its body that allows this straw-like mechanism to function. The animal is able to remove the contents of the hypha to a certain point, and that it needs to move on and find a new section from which you can suck the contents once again. It's actually using its lips in order to find a suitable point on the hypha where it can latch onto. So there's obviously some sort of a mechanism of recognition where a good position is for it to insert its stylet into the hypha, and it will continue to move on and look for the right places as it feeds. But you can clearly see how there is this sucking of the hyphal contents into the nematode's mouth. The good news is that some of the fungal-feeding nematodes can suppress plant pathogenic fungi. So, for example, Aphelencoides and Aphelinchus species can reduce or suppress damping off disease in cauliflower seedlings and other crops that is caused by Rhizoctonia solani. They can also reduce antagonistic mycopathogenic fungal benefits. So that means that some of the fungi like Gliocladium and Trichoderma, pictured here, can feed on some of the mycopathogenic, so to speak, disease-causing fungi. So they already provide biological control, and some nematodes can actually consume these fungal biological control agents. So therefore, the fungal-feeding nematodes can actually reduce the effects or the benefits of these fungi. However, this really depends on the ecosystem, and as with everything that we ever talk about in the soil, we must remember that it is a very complex ecosystem, and when the conditions in it are very favorable to biodiversity, we are not likely to actually have any pathogenic fungi or other pathogenic or parasitic organisms. And usually these kind of interactions occur either in lab experiments or within ecosystems, such as those in heavily managed agricultural systems that are quite unhealthy to begin with. Aphelencoides species grows rapidly on saprophytic fungi, such as Agrosaibi, for example, and also mycorrhizal species, such as Lacaria. And of course, when mycorrhizal fungal species are predated upon by fungal-feeding nematodes, it influences plant nutrient uptake and nutrient cycling. And this could be detrimental, for example, by reducing the mycorrhizal fungal biomass and therefore preventing plant roots being colonized, or stimulate the growth of mycorrhizal fungi. So once again, this depends on the ecosystem and the health of the ecosystem and the predation pressure, as well as the health of that symbiotic relationship between the mycorrhizal fungi and its plant partner, as well as other fungi being present to feed the nematodes. So if there are no other fungi that the nematode prefers, it will actually consume mycorrhizal fungi with greater pressure. So once again, the key is biodiverse ecosystem that supports all the trophic levels where only benefits arise from all the interactions. Aphelinchus species can prevent arbuscular mycorrhizal glomus species and pine root symbiosis, and this is really interesting and heavily studied interaction because pine roots depend on the glomus species to feed the pine trees, and particularly in monoculture situations, and particularly where the pine forest is being farmed, basically, as in the plantation situation, and usually these trees have been introduced into an environment, there are pressures upon these unhealthy introduced ecosystems, which makes absolute sense as nature has ways of actually moving the ecosystem into balance. And one of the ways that occurs is through agents such as fungal-feeding nematodes actually affecting the proliferation of these species, such as introduced pine trees growing in monoculture. Some avascular mycorrhizal fungi trigger aphelincoides species reproduction, and this is a beneficial situation, as this grazing by the fungal-feeding nematodes actually increases plant growth and also has been found to increase arsenic tolerance in arsenic-polluted soils. So these interactions can be quite complex and affect plant growth on various levels, helping the ecosystem to thrive through the increased abundance of avascular mycorrhizal fungi and increase in aphelincoides species population. Predation on avascular mycorrhizal glomus species, as well as Aqualospora species, actually increases avascular mycorrhizal fungal abundance. And this is to do with when there is grazing pressure on the fungi proliferate because they want to regenerate the damage. Also, nutrients become available as the nematode feeds on these fungal species, releasing the excess nutrients and actually supporting further growth of the avascular mycorrhizal fungi. And this is to do with how fast the avascular mycorrhizal fungi can recover from the grazing pressure, and this depends on how fast the fungal feeding nematodes actually proliferate. So there is a balance, and once again, this is an example of a healthy ecosystem where those interactions are favorable. So this is Trichoderma hasianum species, which is known to control some micropathogenic fungi, some of what we call fungal diseases. Let's now look at a couple of examples of what it looks like when fungal-feeding nematodes are researched for their potential as a biocontrol of micropathogenic fungi, or those fungi that are associated with plant disease in agriculture. So in this particular study, Aphelinchus evini and Aphelincoides species were examined for their potential to suppress the tomato corky root disease, which is associated with Pyrenoceta lycopersicii. So soil infested with Pyrenoceta was used, and the treatments included addition of different composts. One compost type was that of green manure, in which red clover was used, plus straw. There was also horse manure and peat compost treatment, as well as garden waste at 70% plus 30% manure and some peat. And the last one was compost consisting of 70% garden waste plus 30% manure and straw. Aphelinchus evini was shown to significantly reduce disease severity at 10% rate, and this is in soil where no compost of any type was added. In fact, nematodes had no effect on compost suppressiveness in any of the treatments, but garden waste compost that contained straw actually did produce disease suppressiveness at a significant rate. However, when nematodes were added to this treatment, the compost suppressiveness was reduced. So the authors wondered whether perhaps the compost suppressiveness was related to some sort of fungi, such as possibly maybe Trichoderma or some other species that are mycosuppressive as well, that actually suppress fungal diseases. And if this was the case, the nematodes might feed on these beneficial fungi and therefore reduce the suppressiveness of the compost upon the Pyrenoceta. The other possibility is also that the compost itself might have had predatory nematodes that were reducing the numbers of the nematodes that were added, but also the nematodes that might have also been present in the compost that would have been doing the job of consuming these disease-associated fungi. So really no one knows what happened there because the compost itself has not been biologically examined to see what lives in the compost and why this particular compost mix was suppressive to the disease. Interestingly, Aphelenchus aveni also lost suppressiveness when treatment with green manure compost was present. So it could no longer function as a suppressive agent in this case. And once again, the question is, what was in this green manure compost that decreased the effectiveness of Aphelenchus aveni? Was it that it chose to consume a different fungal food when green manure compost was added to the soil? This is an unknown as well. Also, interestingly, the horse manure compost actually led to significantly increased disease severity. In fact, this treatment had to be removed from the experiment because it could no longer be studied. And then additionally, Aphelenchoides species had no effect at all. So it just did not suppress the disease in any situation. So this is interesting in terms of what happens when we study things in isolation, but once many variables are present and the ecosystem changes, very different effects occur as opposed to when things are studied in isolation in laboratory conditions. So this is why a lot of studies, even though we are using them here to demonstrate some indication of what is happening in soil ecosystems, they are really unreliable. And I'm always going to go back to this idea that unless we have a very functional, highly beneficial, highly biodiverse food source for all the organisms to function in harmony and produce the most benefit to the plants, we are always going to look for magic bullets. We are always going to look for organisms that are going to help us to suppress diseases that should not even be diseases in the first place. These organisms exist in the soil naturally, and in a healthy ecosystem might it not even ever be a problem and never participate in the symptoms of what we call plant disease. So here is an example of Pyranokita lycopersicii effects on roots when it is in a soil situation that allows its infestation of plants, and in this case it's a tomato plant and it prefers to feed on that, obviously. Interestingly though, this disease in this particular study, it didn't affect the yield in terms of there was no difference in root, shoot or fruit production between all of those treatments. The only difference was that the roots were actually examined based on visibility of disease or occurrence of this effect on roots that you can see here, this browning, corky, unhealthy looking root system. So interestingly, when these authors of this study were initially looking at the suppressiveness of the two different nematodes in the lab before doing the experiment in the field and in the pots in the greenhouse, they found that both Aphelenchus aveni and Aphelenchoides species grew well on Pyranokita. And so therefore they used these two nematodes as potential disease-suppressing agents. However, as we know, Aphelenchoides species had no effect at all and didn't in any way reduce disease severity. So they were asking whether Pyranokita lycopersicii was an actual sufficiently palatable source of food for Aphelenchoides. Maybe when the organism is presented with other food sources, those that would have been in the soil or added with various composts, that Aphelenchoides no longer wanted to eat Pyranokita. And it has been shown in other studies that the two different nematodes actually have different nutritional requirements. So Aphelenchoides has been shown to change food preferences once added to soil versus when it's force-fed a particular fungal food in the lab. And this is the problem once again when we're doing studies. We actually use isolates. We take organisms out of their environment, and we grow them on specific food sources. And even though those food sources might not be their preferential food source in nature, they have nothing else to eat, so they survive on whatever is given to them. So, for example, when grown on agar plates, Aphelincoides composticola does feed on Botrytis cinerea and reproduces, and that's another organism that is associated with plant disease. However, when the species of Aphelincoides is grown on dry root rot fungus, Merullius lacrymans, it does not reproduce. It does not grow. It's not viable while being subjected to this potential food source. And the question is Botrytis cinerea in this case more palatable or accessible to the nematode and therefore a more suitable food choice? So in an ecosystem as biodiverse as soils should be, there will be lots of food choices. And the same goes for these, so to speak, disease-causing fungi. They will not be a problem when there is a biodiversity of food sources for them as well. Different plants available and not just one plant type. And the plants are healthy and resilient and supported by the microbial community to flourish. They will not be susceptible to these organisms that are so stressed in the soil, like they are in the lab, where they are forced to basically eat this tomato plant or consume the grapevine to such a degree that it looks like it's just destroying the plant completely, like is the case with Botrytis cinerea. So it is just a question of the ecosystem health and therefore plant health and plant resilience, where these organisms that are associated with the symptoms of disease, they basically decompose those plants that are somehow compromised or damaged or otherwise unhealthy. And so these organisms are doing their job in decomposing, cleaning up the ecosystem of unhealthy plants so that their biomass can actually be useful for the growth of other plants that are healthy. But in an agricultural ecosystem, all of the plants are impoverished, they're all unhealthy, they're all mistreated. The entire ecosystem is heavily mistreated and malnourished. So therefore, these organisms are there to do their job, and we need to view them as disease-associated organisms rather than disease-causing organisms. The disease is already present. We've created the disease in the ecosystem by mistreating it chemically and mechanically. And these organisms are simply blamed, and more assault happens through the way we treat these organisms as an enemy, using chemical warfare against them. And in this case, we're trying to use a biological approach where we're actually trying to control the organism via, in this case, biological weapon, which is the nematode. However, as we can see, nematodes have their own food preferences and therefore might choose otherwise, just like has been the case with the introduction of the cane toad, for example, in Australia. So here we have Botrytis cinerea, a beautiful image that shows this beautiful organism under the microscope. And none of these organisms should be viewed as something bad, but as something that is responding to a very deprived ecosystem, a very depleted and impoverished ecosystem that is created in agricultural situations through mechanical and chemical degradation of the soil. Another example is that of fungal-feeding nematodes suppressing plant pathogenic Fusarium culmorum. What's interesting about this study is that both springtails, Collembola, the species Fulsomia candida, as well as Aphelincoides saprophyllus nematode were investigated in their suppressive capability against Fusarium culmorum, which proliferated in wheat straw. This is an interesting organism as it is able to produce symptoms in plants ranging from seedling blight to foot rot to ear blight, stalk rot, common root rot in cereals, grasses, and other plants. So it is thought to be responsible for various diseases in various plants. But when it lives in a symbiotic relationship with a coastal dune grass called Lamus mollis, it actually confers salt and drought tolerance to these grasses. So it naturally occurs as a symbiotic partner of plants, of certain plants, but when conditions are favorable, this organism can actually exploit different plants. So just once again, going back to that premise of when the ecology is imbalanced, where there's a problem and the balance is upset in the soil ecosystem, organisms that are normally actually beneficial or go about their business doing good in the soil community can be forced into stressful situations such as agricultural fields to become a problem. But this study looked at the different numbers and combinations of the grazers, so the Aphelencoides nematode as well as the springtail, and the biomass of Fusarium that was present in the wheat straw, and they measured the fungal biomass and the grazer densities that were present and noticed that there was a significant reduction in Fusarium biomass after 2 weeks compared to control treatments. So the authors suggested that it might be useful to study these fungal grazers to control Fusarium by removing it from the wheat straw in the first place, and therefore reduce plant disease risk by adding these grazers into the system. So this is a beautiful image showing the Fusarium pulmorum conidia, which are the asexual spores of the organism. So we have come to the end of lesson 7.5, and in lesson 7.6 we'll be looking at the ecology of the fungivorous or fungal feeding nematodes. But also I'd like to just point out the eye candy that is Aphelincus aveni here in the image on the left from a study from 2022 that points out the anatomy of the species. And this is a scanning electron micrograph depicting this gorgeous creature. So you'll be hearing from me again in lesson 7.6. Bye for now.